Now, before we do anything, okay, so I would actually want to ask you to go to the, if you're using Chrome, all right, go to the setting, all right, of Chrome. And then you need to change the download setting, okay? So if you just go to the setting and in the search panel of the setting, type download. All right, now right away you see here downloads, okay? Locations, you can change that, but make sure you turn on this ask where to save file before downloading. The default, I believe, is off, okay? So change that ask where to save file, each file before downloading, change it to on. That's both safety features because, you know, if you accidentally click on something, it doesn't just download on your computer. It still just, it actually prompts you that you want to save that file, okay? So that's actually an additional safety issue. So, which I believe you should do anyway, okay? So, so turn on this ask where to save each file before downloading, all right? So that it doesn't just automatically download the file. It will, whatever you've clicked on, either by accident or intentionally, it actually prompts you, asks you where do you want to save it, whether you want to change the name before you do that. So that's in Chrome, okay? All right, now if you do Edge, this is Internet Edge, okay? You can also go to your setting and change that, okay? So also do a similar search, okay, in the setting. Downloads, okay, if you, when you see downloads, you actually would see a number of things, okay? Actually, you need to go all the way to near the end here. It said location download. Also, make sure you all the way at the end with Chrome, okay? So with Internet Edge, ask me what to do with each download. Make sure you turn that on. The default could be off, okay? Make sure, always ask me if I want to save a file or open it without saving. Please always turn that on. This is actually a safety issue for your regular browsing too. So, so I said, even when you accidentally click on a file, it doesn't just download automatically. At least it prompts you to say, do you want to download that file? Okay, so this should be on for this exercise, okay? And I would advise you to leave it on all the time for all regular browsing, okay? So all right, so so that's, you can use either Edge or Chrome. I don't know what to do with the Safari, unfortunately. So I'm sure you can find corresponding settings with Safari. I don't have Safari, so sorry about that. But as I said, if you choose to use a Mac, you need to figure out what to do by yourself. Okay, so so all right, so I'm sure you can find similar setting and save it a different, you know, file. Okay, if you use a Safari. Okay, so if you use a Mac. Okay, so all right, so even when you don't, you can still, you know, find the files that you've downloaded and change the names afterwards. Okay, so all right, so now we have the right setting. Let's actually go to this NCBI website. Okay, so which is where we would need to do our inquiry. All right, so this is the NCBI website. All right, let's just copy and paste the over this window. All right. NCBI stands for National Center for Biotechnology Information. It's funded by the NIH, okay, and the US government, okay. And you see on the, there are a number of tools you can use: PubMed, Bookshelf, PubMed Central, BLAST, Nucleotide, all that, okay. Oh, excuse me. Today we are going to use the BLAST, okay, BLAST, nucleotide. You just click on BLAST, and then it will ask you, do you want to do nucleotide BLAST or protein BLAST, okay? We are going to do nucleotide BLAST, okay? All right. Or you can simply click on this link that I've given to you. That should take you straight to the nucleotide BLAST, okay? Which is, you can see here, if you just copy and paste the second link here, it should take you directly to this nucleotide BLAST page, okay? So all right, so everybody got that so far? They have changed the setting. All right, so I'm sorry if you don't have a separate computer, or if you're just doing, if you only have an iPad, you're gonna have to figure out how to do that on a computer later on. You cannot do this on a mobile device. Okay, so it has to. You can only do this on a real computer. Okay, so. The college is actually open. You can go. I mean, you can go to computer rooms. Okay, and then, but you do need a real computer to do this exercise. Okay, so. Anyway, so, and the college still has a laptop loaner program, okay? So, all right, so now you need to copy and paste this human hemoglobin gene, okay? I said under file, open this, but then you can, I've already copy and paste that file for you. So you don't have to download the HBB file separately. You just need to now copy and paste the entire human, this is the human beta hemoglobin beta subunit, okay? So copy and paste that whole ATG to this query panel, okay? So enter query sequence, just copy and paste that onto this whole panel. All right, now a couple of things to do, okay? So now make sure you see at the bottom here, organ program selection, optimize for, make sure you select somewhat similar sequence, okay? I did select somewhat similar sequence. Make sure you do that, okay? Now let's try the first organism first, okay? So you can either type the pen, and then you have to look for the taxid ID 9569, okay? And then, but I think they just changed it back to chimpanzee anyway. Chimpanzee is 9569, okay? Or you just type 9569, which is the taxid ID that I've given you. And then in the organism, you want to type 9596, okay? That will actually give you the chimpanzee genome, okay? So what we're doing is we input the human beta hemoglobin gene on this query panel. We are looking at the chimpanzee genome right now to see if we can find a sequence that is similar in the chimpanzee genome. Okay, so this is what we're doing. Okay, so we will do one species at a time. All right, so we are now blasting the human beta hemoglobin beta subunit gene in the genome against the genome of chimpanzee genome, okay? Now, practically every organisms that mankind have known or have been sequenced, okay? Because sequencing technology is very advanced right now. So you can pretty much look for any species, okay? So we're going to look one at a time, okay? So we'll look at chimpanzee first, okay? So that is widely known as the organism that is most closely related to us, okay? So all right, so if you make sure you select somewhat similar sequences though, okay, that's another thing, okay, so. All right, so now we're ready to blast, okay, blast, okay, so. All right, now as you blast, that can take like, maybe 10 seconds or so, okay, so depending on how many people are doing it together at the same time. If all 20 something of you are doing it at the same time, that's going to be a little bit longer. Okay, so, all right. So one time I was doing with my class and we crashed it because all of us are doing it at the same time. Okay, so anyway, so now this is the output. Okay, you scroll down a little bit on the output here, pen, peniscus, plen, troglodytes. Okay, different species of chimpanzees. Okay, so pen is a chimpanzee. Both actually give you the same output, actually. Identical query cover 100%, and then identity 99.77. Okay, so now in the instructions here, that when I make this was the one that came out. They're actually both the same, okay? You can click on either of those. But let's take a look at this alignment, okay? Pretty amazing. You can see here the chimpanzee hemoglobin beta subunit is very, very, very similar to our hemoglobin with only one base difference, 443 out of 444, okay, matching 99.7%, okay, only one base difference, okay, 443 out of 444. All right, so essentially that means chimpanzees have virtually the same hemoglobin, just one base pair difference, okay, between human and chimpanzees beta hemoglobin subunits, okay, so. All right, so now you can see here, the first two are basically the same thing, okay? You see, it shows you all different types of chimpanzees, okay? Not just one species of chimpanzees, but even the first three are all the same, okay? Different species of chimpanzees here. All right, but we usually, the first one is the best match anyway, okay? So we will just use the first one. So you see here, click on the sequence ID, click on the actual sequence ID here. All right. So pay attention right now. There's a trick to save that sequence correctly, okay? So, all right, so this is the chimpanzee beta subunit, okay? So this is the actual, so what you want to do is to go to right here, send to, click on this little arrow, okay, and then select coding sequence. That is absolutely essential. You do not want complete record. We only want to compare the coding sequence. If you select complete record, that is going to totally screw up your analysis. Please only select coding sequences here. And then you click create file. Now, since I've already changed my setting correctly, okay, that would prompt you to save. Where do you want to save it? Okay. Now I would advise you to create a new folder where you want to save it. Okay. So you see I did it in the morning class, right? So I'm going to do this in the afternoon class again. All right. Create a new folder so that you can save it somewhere safe. Okay. So you can save it wherever you want. Okay. All right. Now you see the file name is just sequence. Okay. Now this is what I really want you to change. Okay. So now if you do not change the prompt to ask you where to save it, it's just going to save it as sequence. Okay. Then you have no idea what sequence belongs to what. Okay. So you should change this to this is the chimpanzee. Okay. Chimpanzee HBB. Okay. Change the name of the file before you save it, so that you know what sequence you're looking at, okay? Chimpanzee HBB, okay? So, all right. We're going to repeat the same process for all the other species. Now, if you do not change the file name before you save it, you're going to end up with a bunch of files that all says sequence, 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 sequence. You would have no idea which one is which. All right. So you need to, when it prompted you to save it, you need to change the file name. All right. Or you need to immediately rename it after it's been downloaded. Okay, so you need to do that. Otherwise, you're not going to know which file name is what. That's why I told you to please change your setting to the download setting. Okay, once again, for you to be prompted under the download setting, make sure you say ask where to save file before downloading, turn that on. I believe the default is off, okay? So if it's off, it will just save it to sequence, okay? Then it's like, you have to find the file, then you have to change the name of the file, which will just make life difficult, okay? So, and then all right, so so this is the, so we've saved the chimpanzee. I put down here, but don't ask me what to do with the Mac. I'm sorry, I don't have one, okay? So I just don't. I just, people, I mean, I don't have a Mac. I don't know how to answer your questions involving Mac. You can make an appointment with Apple Genius. Okay, so you can make an appointment. And if you choose to use a Mac that is not white, I mean, virtually the entire business world uses PC. Okay, it's fine. You use them except for graphic design. Okay, it's fine. You want to stick with a Mac, but you need to figure out what to do. Okay, so just. I'm sorry, I don't have to they don't know what to do with Mac. Okay, you can Google it or make an appointment with Apple Genius, please. Okay, so anyway, so that is one species. Okay. Now we would like to repeat the same process with all these species, okay, before we construct a phylogenetic tree. Okay, so actually I would advise you to close this window so it doesn't obstruct you. Go back to the original blast window, scroll back up, okay, and then click on it, edit search. Okay, you see the human hemoglobin gene is still there. And then now we just change the species. Okay, so next one I want you to enter is some New World monkey. Okay, monkeys 9479. Okay, so I've already given you the text ID. You can just type in the text ID. Okay, so all right, and then do the same thing. Leave that as somewhat similar sequences. Now we blast. Okay, so. All right, so other monkeys now. Okay, this is Lacothrix leukotracha. Okay, so let's see what monkey that is. Okay, all right, this is I'm just curious what monkey this is. Lacothrix leukotracha. This is the pretty cute monkey. All right, this is likely to be the Leukotracha, the woolly monkey. Okay, brown woolly monkey. Pretty Pretty cute. This is Leukotrix leukotraca. Okay, so that actually turns up to have the closest homology. That's a different species. All right, actually the same identity. All right, and then so usually the first one is the best match anyway. Okay, so I just click on the first one. Leukotrix leukotricha. This is the brown woolly monkey. Now you can see that the identity dropped to 420 out of 444. Only 95% match now. Okay, there are a couple more differences. Okay, so in the human hemoglobin gene versus this brown woolly monkey. Okay, Leukotrix leukotricha hemoglobin. Okay, so you can see here, now this is chimpanzee. Everybody knows that this is the organism closest to human. If you go to another kind of monkey, another primate, you can see that there is a drop in the similarities. Okay, there's now a 95% identity only. Okay, in terms of the in terms of the hemoglobin gene. Okay, all right. So you want to do the same thing. You want to click on the sequence ID. All right, and then go to send to. All right, so click on coding sequence and then create file. Okay, so, and then this is our brown woolly monkey. Okay, brown woolly monkey. Actually, yeah. Make sure you change name before you save it. Okay, so otherwise you're not going to know what species you're looking at. Okay, so. All right, so same thing here. Close the window. Okay, and then go back to the original last window, scroll back up, click edit search. Now let's blast it against the dog genome. Okay, so 9615, beagle dog, canis, is probably all the same. So it doesn't matter. All right, so or just you type dogs. Okay, that's generic dogs. Okay, 9615. Okay, so that would include all the dog species. Okay, so. All right, so dogs, okay, so let's see if we find a similar hemoglobin in dogs, okay, so well, dogs have blood, obviously it should have hemoglobin. We'll just wait for the result. All right, now in the dog, Canis lupus familiaris, all right, this is our dog. You see the identity of the hemoglobin compared with the human ones now is only 87%, okay? So very evidently, as you go to species that are less related to us compared with the monkey, the dog have a much lower level of similarities now, only dropped to 87%, okay? It's only matching 385 out of 440 now, okay? 88%, okay? Compare with the chimpanzee one that is 99.7%, and the brown woolly monkey, there was 95%. The dog has a significant drop in the similarities, okay, between our hemoglobin, okay. So, all right, so click on the sequence ID again. We want to save that sequence. Once again, go to send to coding sequence only. Make sure you save only coding sequence, otherwise it will be, this is dog, okay, dog HBB. All right. So close that window, do the same thing to other species. Okay, so now I want you to download all these species plus 2 more of your choice. Okay, so let's go to the cat right now. Cat family, cats, 9685. Okay. All right, now let's blast it against the cat genome. Let's see what we come up with in cats. All right, cats, Felis cactus. Okay, also beta subunit, Felis cactus. All right, usually the first one is the best match, except there's a snafu right down the middle. We'll get to that when we get to bats. That's a. All right, Felix cactus here. Same thing we've been doing. Click on the sequence ID, send to, make sure you download only coding sequence, create file. This is cat. Make sure you change the name correctly. Cat, okay. All right, so cat, you see, we're seeing, what's the percentage similarities in cats? In cats, the similarity is 86%, okay, a little bit less than a dog, okay, so the dog seems to be a little bit closer to us compared with the cats, okay, so I think dog owners would like that suggestions. Okay, so dogs are somewhat evolutionarily closer to us than cats. Okay, so you see 86 versus the 88% we find in dogs. Okay, so all right. Now let's go to a less related species, mouse. Okay, so 10088 mouse. Okay. All right, so now we're going to blast it against mice. Okay. Yeah, somebody's Andrew said you have your iPad. I'm sorry, you can't really do this on your iPad. Okay, so you have to have a PC, a real computer to actually save the sequence, and we'll upload it for alignment later. Okay, so unfortunately, iPad is still not. Right, doesn't work for all situations.
So if we don't have access to a PC, we just have to figure it out then.
I'm sure you don't have any computer access anywhere.
No, I have a MacBook, but it wouldn't
I
be
mean, MacBook, you can do the same thing with MacBook. It's just that I don't know how you can change the file name before you. I'm sure it's saved it somewhere, and then you just have to rename the file and dig out a sequence later on. Okay, so,
Okay.
so yeah, I'm sure you can do the same thing in Mac. Okay, so I don't have a Mac, so I can't tell you how to do it. Okay, so that's just my point. I don't have a Mac. I don't exactly know how to tell you what to do with a Mac, but. And I don't believe that's my responsibility. Okay, so I'm sorry. Okay, so the whole world uses PC, and then it's like, if you, I mean, I can't basically support every single platform. Okay, so I mean, if the college give me a free Mac, I may learn how to use it, but I don't have one. Okay, so anyway, so this is the mouse one. Okay, so you can do similar things in Mac. Okay, it shouldn't be. I mean, you can go to the browser and the sequence will come. It will let you save it somewhere. Okay, so. And then the question is just rename the file correctly so that you don't mix them up at the end. Okay, so all right, so human hemoglobin here. Okay, so this is the mouse. Okay, so we are down to mice right now. Okay. All right, so okay, so you see here, this is mouse hemoglobin. All right, so if you take a look at that, the similarity dropped down to only 83%. All right, so not surprising, the mouse is a lot less related to us, okay, body and form compared to, even the cat and the dog and monkey for sure. So you're seeing a lower percentage identity here. This is the mouse one. Coding sequence only, once again. I mean, the college has the computer room in the college is open. You can, if you really need a PC, the college has plenty of PCs, and then I'm sure you can figure out how to do it on a MacBook. Okay, so it's like just changing the name of the file. Okay, so it's just, and all right, so so now, okay, we've done the mouse right now. All right, so let's just go to other species. Now the bats that I've included, there's a little bit of attention. You need to pay attention to the bats. Okay, so when we change it to bats, okay. All right, so when you blast it against the bat genome, there is a wrong entry in the database. I don't know, that didn't used to be the case, but now there's a, all right. This is bat. Now the first one in bats, this one, whatever's the oriental, this is actually not the right entry. You see it, 99.77%, that's the same thing in a chimpanzee. Okay, so the first one in bats is actually not, that's not a bat. Okay, so this is the one that you find in chimpanzee. Okay, so somebody mislabeled it and put the wrong information on the NCBI database. So since it's quite obviously this is the chimpanzee one, okay, somebody just thought that as a bat. Okay, so I don't even know what this species is. Okay, so this is the wrong one that you do not want. Yeah, I mean, yeah, so that's the wrong. That's definitely, this is the bat, but the chimpanzee one, this is clearly a chimpanzee one, 443. So please don't use the first one that you see in this bat. Okay, so let's go to the second bat. Okay, or the third bat, vampire bat, Pterops vampirus. Okay, so this is actually a better bat. Okay, vampire bat. Okay, Pteropus vampirus. Okay, vampire bat. We know for sure this is a bat. Okay, so all right. So now you see with the bat. Okay, wow, 89% match, 393 out of 442. Even higher than the dog, higher than the cats. Okay, so not as much as the monkey. Monkey was 95%, remember? But you see with the vampire bat, the identity is 89%. Okay, so you see the bats are actually closer to us than the dogs and the cats. Okay, so from at least comparing the sequence of the hemoglobin. All right, and that's probably why we get viruses from bats. Okay, and not less likely to get it from dogs and cats. Okay, so and then that's why we're getting probably why one of the reasons why they are closer to us phylogenetically. Okay, compared to even cats and dogs. Okay, so all right, so a vampire bat. This one looks good. All right. Now, as I said, please ignore the first entry that you see in bats. That's clearly the chimpanzee one. Somebody made a mistake, maybe just naming a species wrong and putting the database correctly. Okay, the database is only good for, you know, whatever people have put into it. All right, so vampire bat. Okay, so I would advise you, when I make this worksheet, that wasn't a problem. Okay, so and then all of a sudden some people put a wrong bad entry into it. Okay, so please use the vampire bat. Okay, Pteropus vampirus. All right, so let's save to. All right, so this is VBAT. Okay, I'm gonna go that vampire bat. All right. All right, so after that, I don't think there's a problem with the rest of the species, but let's actually do those. All right. All right, so let's try pangolin, the other known host to COVID-19. Okay, so I put it in there. Pangolins. There are two pangolins. I don't know the difference, but let's stick with the 9971 anyway. That's the one that I've used before. And then blast. Okay. All right, with pangolin, okay, 86%, okay, very close to the cat, okay, so pangolin, all right, so 86%, quite similar to the cat's figure, okay, so, and then, all right, so this is our pangolin hemoglobin. Ooh, we will save that sequence too, coding sequence. I think the only problem with Meg is that when you click create file, you may save it somewhere. Okay, so that's probably the only difference. When you click, it may just download it somewhere. You just have to go and rename the file and find it. Okay, so it's really not a problem. Okay, so the only problem you encounter with Mac is that when you click create file, it may save it somewhere. Just go find it and change the file name. Okay, then. All right, so that's probably the only problem you experience with Mac. Okay, so because I don't know how to show you how to, you know, I don't know what settings you change to that allow you to change the file name, but it's just, I'm sure you can solve it with Mac very easily. Okay, so all right, so this is the pangolin. Okay, yeah, the only problem with Mac, when you click on it, it saves the file somewhere. Okay, so you probably just have to immediately go find a file and just change the name. Okay, so that you don't get you know, you just have to do those, figure out how to do those extra work yourself. Okay, so all right, so this is the pangolin. Okay, so let's go to something really less related, like koalas. That is a marsupials. That is, it's still a mammal, but it's a marsupial. Okay, marsupials are only found in Australia, and then conceivably that would be more different from us compared with other mammals. Okay, so koalas. All right. Okay, so this one, 78%, all right? So subunit epsilon, okay? All right, now you see here, remember all of the other mammals that we look at, even the mouse is 83% or so. Now with the koalas, that's a marsupials, we drop down to 78%, okay? So that is not surprising. Marsupials are much less related to us compared with other mammals, okay? So that's only found in Australia. They clearly probably diverged from the rest of mammals over 150 million years ago. And then with all this time elapsed, obviously this hemoglobin has also changed. Okay, the blood of a koala is conceivably would be more different to us compared with other mammals that are closer to us. Okay, so all right, this is a marsupial, a koala. You can find kangaroos. You can find pretty much all other organisms on this website. Okay, so all right, coding sequence, create files. All right, now let's look at something much less related to us: chicken, salamander, snakes, and fish. Okay, let's see how those stack up. Okay, so all right, go back to this. Click edit search again. Just change the species. That's the easiest thing. All right, chicken, chicken, 9031. Okay, so all right, chicken, Gallus gallus. Right, so all right, we have one that is about 75% match. Okay, so all right, that looks like the right one. 75% match. Okay, you see, as we go to species that are less related, you can just look at the percentage. You can already tell that it's less related to us. Okay, so it's already kind of telling the evolutionary story just based on how related the species is to, with respect to each other. All right, so you can see coding sequence, create file, same thing, chicken. Okay. All right, so I'm just going to quickly download the next three. Okay, so we can move on to the second part. So we can actually do make the tree. All right, so chicken, salamanders right now. Okay. Salamanders is a amphibian. Okay, so let's see if it's more or less related to us compared with a chicken. With a chicken. You can probably guess it's going to be a little bit less. Okay, so all right. There's only one homology in the larval, 68.55%. That's actually the limit of where it can find similarities. Okay, so all right. So this is the all right, same thing, coding sequence, create file. This is salamander. All right, let's try the snake and the fish. Okay, so we're gonna do snake. All right, so now we have snake. There are different species of snakes. Okay, 70-something% identity. Pseudotus textilis. This one a little bit higher. Now, even though this one has high similarity, the query cover is less. Okay, so I put a little note here. Make sure you select the one with the highest query cover. That means it's, comparing the maximum length of the gene. Okay, so usually the first one is the best match anyway, except for the one snafu in the bats. Okay, so all right, this Pseudonaya textilis, that sounds like a snake to me. Coding sequence, create file, snake. Okay. All right, let's go to some fish. Now you can play with the fish. There are many different types of fish, okay? If there's some kind of fish you can think of, okay? So, such as salmon, okay? So, king salmon, all right? And then instead of using that bony fish, you can choose a specific kind of fish, okay? So, I'm going to just a little variation to king salmon, okay? So, just because I like to eat them. So, you can stick with the just fish, but you can choose any kind of fish you want. Okay, I just want you to put in a fish there so that just for comparison. Okay, so all right, salmon back to 68.9%. Okay, cool. All right, now I said here, please add 2 extra organisms of your choice. Okay, don't just copy my tree. After you've saved all of these organisms, add 2 more of your choice. Okay, when you submit your work, and then let's see, what do you want to do? Let's say what organisms looks fun. Okay, let's throw in a whale, orca. Okay, let's put a little marine mammal. Don't copy my choice. Think of any other organisms, horse, cat, whatever. I mean, lions, whatever. Okay, so all right, so okay, whales. There is one here. 99%, 87. Oh, whales quite close to us too. Okay, so you see. There are a lot of these marine mammal, but it's quite close to us, apparently. More so than the dogs and the cats. Okay, so that's whale. All right, so let's throw in one more. Let's try. Well, let's change things we can try. We can try. How about just a cow? Cow. All right. All right, that's the cow one, 86.9. Okay, so all right. All right, so and I've downloaded 13 altogether. Okay, so I have human, chimpanzee, and then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. We had 2 more, so 15 altogether. Okay, so not a lot to begin with. You can add more if you like. Okay, so feel free to add several more. Okay, so if you all right, so let me just close this window now. All right, now you need to open you can read the instructions going. Now you need to open a blank Word file. All right, you can do it on Notepad, you can do it on Microsoft Word or whatever word processing software you do. Okay, so now put in the human one first. Okay, so you need to do is to put a little forward arrow, middle bracket. Let's put in the human one first. Okay, for the human one, you just need to copy and paste as the human one that I have downloaded for you in the beginning. All right, now let's go to the folder of where I've saved all of those. Okay, so I save all of those files in the spring 2001 afternoon session. So this is where you need to be careful, find out where you've saved your files. Okay, so actually this is the morning session, afternoon session. Okay. Okay, so we downloaded monkey, cat, chicken, chimpanzee. You see, that's why I do this. So now I'm very neatly organized. Okay. And then I said, if you use a Mac, you're likely to get sequence 1, sequence 2, 3, 4, 5, and then you have to just change the file before you actually change the file name before you save it. Or maybe after each file, you just need to save it individually. Okay, so, and then anyway, all right, so now I actually think it's good to do it just alphabetically. Okay, so it's sorted alphabetically anyway. So I'm just going to click on the first one. You see that appear, opens in a notepad. And then there's a lot of junk in there, right? So it's this ICOD sequence ID hemoglobin. And then this doesn't have the species name in there, okay? So if you do not save it as a different, if you do not change the file name, you are not going to be able to tell what species that is, okay? So it's just, that's why I said it's important. Before you save the file, change the name, okay? Or after you save the file, immediately change the name, okay? So you know which species is which right now, okay? All right, so here's what you need to do, okay? On your word processing browser, on your word processing file, this is brown woolly monkey, right? So brown woolly monkey. All right. All right, and then now you just need to copy and paste from the ATG on, all right? Why does all the gene begin with ATG? Anybody remember why? Every single gene here that we've downloaded begins with ATG. Why is that the case? What is special about ATG? Anybody remember? All right. Take a look at the cat one, begins with ATG too. All right, so this is the cat one. Okay, anybody remember why every single gene begins with ATG? All right. What's special about ATG? What is the... All right, so you see you open different species. Make sure you only copy from the ATG, okay? Just take away all those junk sequence, okay? Otherwise, you're not going to be able to do a valid comparison, okay? So cat. All right, now you need to combine every single sequence that you've downloaded into one big file, okay? This is what we're doing right now. I would advise you to do it alphabetically, so that's easiest, okay? So, and then the program will align it correctly anyway, okay? So, and then make sure you only copy from the ATG. I still haven't heard an answer. Why does every single gene begin with ATG? What is special about ATG? What do you call that codon? DNA, what do you call ATG is what codon? Okay, so ATG is every single gene begins with ATG. Like the start codon, yes, thank you, somebody. That's universal to all life forms, okay, not just the human. ATG is the universal start codon to all life forms, okay. So, all right, so now I'm up to the chimpanzee here. Chimpanzee, okay. So that's why please make sure, just, when you open the text file like that, it shows you a lot of junk. Only copy and paste from ATG. Now, it could be, you can screw up here easily, okay? If you mix up different genes, then your tree will look very funny, okay? So make sure that you carefully open each file. You see, I close it before I go to the next file, okay? So you don't mix up the different species, okay? And, all right, so this one is a cow, okay? So I would do it carefully one by one because it's easy to mix up, okay? If you leave too many files open, make sure you copy only from the ATG of each species and put it under the right heading, okay? That's more important. Put a little forward arrow, put a little middle bracket here, and then type the name of the species, close the bracket, then put an enter, and then immediately paste the sequence next to it. When I'm done, I close it, before I go to the next one, so that I don't mix up the different species. Okay, so all right, now I'm done up to the dog. All right, so you get the idea what to do. I'm just going to try to copy and paste it quickly. All right, so this is a demo. If you can catch up with me, you can do it. I'm sure someone is doing it faster than I am right now. And then if you need a little bit more time, you can definitely go ahead and do it yourself later. All right, and then I'm up to the koala right now. All right, close that. Now let's go to the mouse. Okay, see, I just close each one after I've mouse. Always just copy from the ATG. All right. So you have to do this, otherwise it will. It's not going to. The program is designed that it just it reads as a name, then it compares the sequence. All right. So pangolin now. All right. Copy from the ATG again. The next one, salamander. All right. Close it. Then I'm going to go to the salmon right now. You can try any fish, okay? I mean, I like salmon because I like to eat them. All right, so, all right. There's a king salmon. I'm going to put a K in front of it. All right, so snake. All right. All right. Now our vampire bat. Okay. All right. All right. My last one is the whale. All right. All right, so now we have created, okay, now this is what you need to create based on all the sequences that you've downloaded, okay? So open each file individually, copy and paste over the sequence, but give it the correct heading, okay? So that's what the program needs to read, okay? So that's the work that you need to do, okay? So you just need to open each sequence individually, okay? I put down the same instructions here, okay? So, put pen, human, dog, cat, okay? I did alphabetically this time, but it doesn't matter what order it goes in, okay? So the program just aligns everything, okay? So, all right, good. I said don't include organisms that you don't find any homology, okay? I did put down a little in the middle. If you try sharks, they don't seem to have any homology to the hemoglobin, okay? And octopus earthworm, you're not going to find homology, okay? So yeah, so it's just you're just not gonna because they don't even they have different kinds of blood. They probably sharks I think have blue blood. They have hemocyanin instead of hemoglobin, okay? So all right, so okay, now with this big file in place, what you need to do is to go to this website, okay? So called the cluster omega. Now, if you don't have time to finish all 12 species, you can still try it, okay? Just 3 or 4 to try it out first. Then you can do the whole 15 species to do the complete analysis, okay? So, all right, so you need to open the browser again. All right, go to this website, okay, the Cluster Omega website from the UK. All right, I couldn't find an American website that makes me do this, that lets me do this without pain. So anyway, this one works, okay, so. All right, so so now it says multiple sequence alignment. That's the Cluster Omega. It's a pretty multiple sequence alignment tool, free online. Okay, so now we are comparing DNA. We're not comparing protein, so make sure you change to compare DNA. All right, so we're comparing DNA. We're not comparing protein. All right, so this sequence in any supported format, just copy and paste this entire big Word file, all right, into what you've made here. Copy and paste this entire thing. All right, copy, paste. So I use Control C and Control V as the command, but you can also right click. Okay, so you can say copy and paste. Okay, so all right, but that is identical in Mac. Okay, so the Control C, Control V is identical in Mac. Okay, so all right, so just paste the entire query sequence. Okay, the entire file that you just made. You can check, see if you've done it correctly. Human, cat, and unruly monkey, cat, okay, chimpanzee, all that, okay. All you need to do now is to click submit. Depending on how many people are doing at the same time, it actually usually gives you the result quite quickly. Oh, right away, great. So it's aligning, you see here, the sequences of all these different, of these 15 species that we've entered into it, okay? So it's basically comparing, all the sequences, okay? So doing a over alignment like that, okay? All right, so you don't need to pay attention to the actual alignment that actually differs, but just click on the guide tree, okay? That should give you a lot of interesting information here, okay? So, all right, that is the same concept of what Darwin was just constructing. Remember, we were talking about Darwin's trying to construct, phylogenetic tree, right? How organisms are similar to each other, which one are more related to each other, okay? So now, obviously, human and chimpanzees are the closest to each other, okay? So, all right. So now the way you read the tree is that human and chimpanzees, from the interpretation of this result means we are most closely related, means we share a common ancestor. And then the next one is monkey. Monkey is kind of, the woolly monkey is obviously related to chimpanzee and human. And then, so that the branching pattern here means, okay, the monkey and the ancestors of human and chimpanzee share a common ancestor. All right, so human and chimpanzee share a common ancestor. And then the brown woolly monkey and the ancestors of human and chimpanzee share one common ancestor. All right, so this is what this branching means, okay? And that actually share a common ancestor with bats, okay? So that is interesting. You see, the bat is actually closest to us compared with dogs, cats, pangolin, all the other things. All right, so we can already tell from the percentage identity. Remember when we downloaded hemoglobin with bat, it was 89% compared with the 87% in dogs. So not surprisingly, the bat is actually closer to us, okay, compared to closer to the ancestor. From this analysis actually showing us that bats actually share common ancestors with primates, all right, with these all three primates, okay, we actually share a common ancestor before we share a common ancestor with the dog, okay, so actually pretty cool. All right, so, and then the cats and the dog, cats and the rest of all these share one common ancestor, then the pangolin, then the whale and the cows before we get to the mouse, okay? So this is interesting, okay? So whale and cows, whales and cows are closely related. That is interesting. That is not expected, okay? So whales and cows are more closely related to each other than, and then the mouse. Mouse should be, one of the earlier primates. Mouse is an early mammal. Okay, so clearly it share common ancestors with the ancestors all of the other ones. Okay, so that's less related. And marsupials obviously is it's found in Australia only, so it's still mammal, but it is less related to us, okay? So we last share common ancestors. So all mammals, all the placental mammals, all of the rest here are placental mammal, okay? You see it forms a very good natural grouping. The mouse all the way to human, we are placental mammals. We have a placenta. Marsupials don't use placentas. Marsupials are like kangaroos. They're born very, very prematurely, and then they have to cling onto a milk pouch to drink milk from a very, very early on, okay? So, and then, but we are both mammals, so we actually share a common ancestor, okay? Before we share common ancestors with the chicken and the snake, okay? So chicken and snakes, they are both descendants of dinosaurs or reptiles, okay? So definitely they're more closely related to each other compared to the rest, compared with other mammals. And interestingly, this amphibian and the fish, okay, fish is likely to be the earliest ancestors of all land vertebrates. So, and then you have the salamander, amphibian, and then you have the reptiles. Okay, this is a very good evolutionary tree. Okay, so this tells you how organisms are related to each other. You can also take a look at this phylogenetic tree, okay, which gives you presented data somewhat similarly, but same idea, okay? Human, chimpanzee, and then monkey, bat, okay, and then cows and whales share common ancestor, cats and dogs share common ancestor. I mean, it's just a different way of presenting, but I like the guide tree more, so it actually shows you which one's more earliest, which one's kind of like which one diverged from one earlier. You can either give me the guide tree or phylogenetic tree, doesn't matter. So I do want to ask you to save this picture as a PDF. So in PC, very easily you can do it. Click Control P is print, okay. And then save as PDF. You can choose your printer, you can save as PDF. If you're a Mac, you can just take a picture. You can submit a JPEG, that's fine. So make sure it's not my tree though, okay? So I will notice that you're submitting my tree, right? So save it as PDF, okay? So, all right, so this is the HBB phylogenetic tree. All right. In PC, you can simply right-click on anywhere on the screen and click print, and there's a default save PDF, okay? So if you don't know how to do that, just take a picture and submit the JPEG, okay? So, all right, so where do you submit it? You go back to your lecture canvas now, okay? So you actually have this window open here. All right, so I like all the grades to be in the lecture canvas since it's a counted grade. So you